Thank you. I'm going to talk about how we can use AlphaFold models for prediction in terms of variance and the effect on structures. Why do we want to do this? So we know that there are very large sequencing projects that are ongoing and are trying to characterize not just our own genome, the human genome, but also the genome of other organisms. And these large sequencing projects are generating millions of genetic variants. And now the big challenge is to understand this genotype-phenotype relationship. Why is this important? Because once we understand the genotype-phenotype relation, we can understand better the biology of an organism. We can also understand better mechanisms that lead to disease. We can formulate better genetic diagnosis. And when it comes to us, humans, we can also, this could also aid us to obtain this personalized medicine, so specific personalized treatments. Over the last 20 years, there have been several in silico tools that have been developed to predict the effect of missense variants, so those genetic variants that cause an amino acid substitution at protein level. As we can see here, we have several, many of them are individual predictors, some of them are meta-predictors, but as we can see, they are all extremely good in terms of accuracy, and they can almost all of them give us this binary outcome. So they're very accurate in telling us whether the variant is predicted to be damaging or tolerated. In some cases, there will be some uncertainty in the predictions. But even if they are all quite accurate, very accurate, most of them, if not all of them, do not give us a mechanism, do not explain us why some of these variants are predicted to cause disease or to be damaging. If we do have a structure of our protein of interest that harbors a variant, we can then start analyzing the structure. We can identify all those chemical physical bonds that keep the structure correctly folded. And we know that one of the main things that we need to have a good functionally protein is a good folded structure. So a few years ago, we generated an algorithm that takes just protein structure, so no sequence information, just using protein structures, it can predict the effect of a variant. What it does, very simply, this algorithm that we've called missense 3D, it takes the wild-type structure, it performs an in silico mutagenesis by removing the side chain of the query residue that harbors the variant. It also removes all the side chains of the environment, the 3D environment around the query residue, and then uses the squirrel algorithm to repack the structure after we have inserted the side chain of the mutant. After we have done this in silico mutagenesis, we now have a wild type and a mutant structure, and the algorithm automatically performs a structural comparison to see if the variant causes some misfolding, identifies any of many possible structural features that can be identified, like disruption of bonds, clashes, cavities, and so on. When we look at the structures that are available, as we have heard over the last 2 days, we have millions of structures, not just the ones that are experimentally deposited in PDB. We now also have AlphaFold models for millions of structures for many, many organisms. When we look at our own proteome, the human proteome, if we consider experimentally generated structures, but also AlphaFold models that are accurately modeled or are ordered and have this PLDDT hard threshold of 70, the coverage of our proteome is around 60%. What we then wanted to do with this project is to develop a pipeline to automatically identify those amino acid regions that are accurately modeled by AlphaFold and that can be used to assess the impact of a missense variant on structure. So what we did, we obtained a large number of amino acid substitutions from Nomad, ClinVar, UniProt, and also all the AlphaFold models for the human proteome. From this large data set, we obtained a benchmark data set. We only wanted variants that have a clinical annotation of benign or damaging, so we excluded everything that has no annotations in ClinVar or has an annotation that says that the variant has an uncertain clinical significance or conflicting annotations. We work with over 89,000 variants distributed in 10,000 proteins. We split this data set into training and test. And then we further split our training test into 5 subsets. We wanted to make sure that there was no homology leaking between training and test, or at least we wanted to minimize that. And we also didn't want to have leaking between the 5 subsets in the training test. And therefore, we used the MMSEC2 algorithm to look for protein homology. With AlphaFold, we know that AlphaFold produces one single model for proteins that are less than 2,700 amino acids. So everything larger than that was not part of our benchmark data set. For each of the over 89,000 variants in our benchmark, we perform an in silico mutagenesis. So we obtain a wild-type structure for the protein harboring each of these variants. And then with our in-house engine, missense 3D, we generated a mutant structure for each of these 89,000 variants. And then we performed the structural comparison between wild-type and mutant, and looked for the 16 features that we can see here on the left. Then we wanted to, in order to identify the regions that are, let's say, accurately modeled that we can then use for the structural analysis, we use the PLDDT. So AlphaFold tells us that the PLDDT gives us this hard threshold of 70 to say that the residue is accurately modeled. So we took, of course, that one into consideration. We call it the PLDDT for the one residue. But then we said, what if we look at the context in which this residue N variant occurs? And so we looked at the average PLDDT in the linear sequence around the amino acid with the harboring the variant. So we look at the window in linear sequence of 5 amino acids. And then we expanded this to 13 amino acids. And then we expanded this to 31 amino acids. And these are somehow some threshold that are arbitrary threshold. For the 31 residues, we selected this because it could represent the minimum length of a small domain. But then we said, okay, what about the 3D space? What if we start looking at what happens around the variant of interest? And so what we did, we calculated the average PLDDT within a 5 angstrom sphere around the variant of interest. And we called this the PLDDT 5 angstrom. So some results. When we look at, so when we use the PLDDT 70, so this hard threshold from AlphaFold for the single residue that harbors the variant, and we calculate the ratio between the true positive and the false positive rate. We bind all our variants according to the PLDDT. We can see that when we reach this 70 threshold, there is suddenly a sharp increase in the ratio, in the true positive, false positive ratio. What about the other scores that we selected? When we look at the linear amino acid region, so whether it's 5, 13, 31, we can see that we have this sharp increase in the ratio, in true positive, false positive ratio, when we look at an average PLDDT of 60. So we can lower it to 60. And we have similar results also when we look at the average PLDDT within the 5 angstrom sphere. Again, we have this sharp increase in true positive, false positive ratio when we are in this bin. So average PLDDT of 60. What does it mean? So some more results. First of all, there are a few things that I would like to point from these results. So first of all, you may all say, well, sensitivity is low, around 35%. Yes, but we need to remember that we are looking at all possible missense variants in humans that have been associated with disease. Here we are only looking at structural damage. So we know that a variant can be damaging, can cause problems to the function of a protein, not just because of a structural problem, but there could be other reasons that we are not capturing with this method because we are not using any sequence information. And we know that if we use other algorithms that are purely structurally based, we get a similar sensitivity. However, we have a very high specificity, precision, accuracy around 65. Of course, this is also dependent on sensitivity and specificity. So the first take-home message is that whether we are using this hard threshold of 70 for the single residue, or we use the average PLDDT either in linear sequence or in space around the variant, we have comparable results when we look at sensitivity, specificity, precision, accuracy. So where is the difference? The difference is actually here. The difference is in the number of residues for which we can make accurate predictions. And we go from an average of 65% of residue when we use the hard threshold of 70 to around 70% of residues for which we can make predictions when we go down to an average PLDDT of 60. So you're all saying, well, it's only 5%, tiny number. Yes, but when we put things into context, 5% of 9 million residues just on the proteins that are less than 2,700 residues means an extra of 500,000 residues for which we can make structural predictions if each of them were harboring a variant. And so we look at all the data. We had this very large data set, millions of variants in our human proteome. We used our in-house engine, missense 3D, and we performed a structural mutagenesis with structural prediction for over 6 million of these variants. Those are the ones that we could map to AlphaFold models. We put all the results into a dedicated database, and then we made all the results available, all these pre-calculated results available in a new resource that we called missense 3D AlphaDB. So it's a database of predictions generated with our in-house missense 3D engine using AlphaFold models. And these are the kind of results that we got when we analyzed all this data. So first of all, we can look at the results on all the variants, and we are looking at over 6 million human missense variants distributed in over 16,000 proteins. These are the sort of small proteins, less than 2,700 amino acids, for which we have one single AlphaFold model per protein. So when we look at all the variants, what we can see here is that if we compare the numbers, so first we use the hard threshold of 70 for the single residue, and then we compare the results with an average PLDDT of 60 for all the amino acids that are in space around the query, so not just the query residue. We are looking at 347,000 extra variants for which we can make predictions, accurate predictions. These are all variants with, the majority of them, millions of them, with no clinical significance. So we don't know if they are benign, damaging. We have no data. They have just been identified in humans, in the human population. And what our analysis shows is that approximately 15–16% of them are predicted to cause some damage to the structure. What if we start looking at the minor allele frequencies? So what if we consider the variant and the structural effect in terms of how common or rare the variant is in the population? And for this, we used the minor allele frequency in the NOMAD database. We divided the minor allele frequency to have the set of common-ish variants, those with a MAF greater than 0.1%, and the rare variants less than 0.1%. We can see that still here, we still have around 10% of these variants, some of them will be quite common, that have some structural damage identified when we perform the structural analysis. And when we go towards, in the realm of the rare variants, this percentage increases. Then we looked at ClinVar, and we were particularly interested in looking at the VUSs, so those variants for which we have no clinical annotations. They are variant of uncertain clinical significance, and these are the ones that pose problems in clinics because we don't really know what they do. And again, we can see that, first of all, if we lower our threshold and use this 60 average PLDT in the 3D space, first of all, we can assess 13,000 more VUSs. And again, we see that there is quite a big number of variants of these VUSs that are predicted to cause some structural damage to the protein. So they could potentially be disease-causing. So we, as I said, we put all these pre-calculated results into this new resource that it's already available from the missense 3D portal. This is our portal with all the resources we've developed to predict the effect of variants, missense variants. The missense 3D AlphaDB can be searched using the UniProt ID or the gene name of your protein of interest. And this is the result page, for example, for the growth hormone receptors. So the database can be searched according to PLDDT, according to damaging predictions, confident predictions, and so on. And then, of course, the strength is the structural analysis. And we provide, when clicking on this link, we provide a specific page with all the detailed structural analysis. Of course, we could do all this on millions of variants because we have this in-house engine called missense 3D, and we just released the version 2 of the algorithm that now allows the users not just to query and make predictions using protein structure, proteins and structural information, but also using genomic, so genetic information for the variant. And these are the input page. We perform a structural check. And then, of course, there is a result page with the detailed analysis. And these analyses can be performed on the single chain, but also on complexes using experimental structures, structures from AlphaFold, and also structures derived from homology modeling. And just to conclude, why do we still want to use homology modeling? Well, while we were doing this analysis on large data set of variants, we saw that sometimes there is a discrepancy between the numbering of the variant on UniProt and AlphaFold models. And so we ran a proper analysis that is now available in bioRxiv that shows that the AlphaFold models that we already have in the database, in the AlphaFold DB database, are already not in sync with UniProt. And this is quite significant when we look at organisms for which, there is a ongoing curation, and it's small. It's only 2.8% for human and mouse. So just to conclude very briefly, what I hope I have shown is that by lowering the PLDDT to 60 and looking at the average PLDDT score in 3D space, we have results that have similar accuracy, but we are able to predict the effect of over 500,000 additional variants. These variants are all available in this missense 3D AlphaDB database that I just presented. And overall, the take-home message is that structural predictions can be made for many variants, and they are complementary. So we can use them with other information that come from other tools and sequence-based methods to predict the effect of variants. Thank you.